Hello, everyone. Welcome back to the WebEVTalk series on the 19th October 2021. And today is a special day. We have two speakers from the industry who's going to share their knowledge with us. The first one is Olga Shatnyeva. I hope I spell your name right. And the second one is Andreia Silva. So Olga is from Cell Therapy Evotec in Germany, while Andreia is from Discovery Sciences, AstraZeneca in Sweden. Olga is a biologist with an academic background in immuno-oncology and extracellular vesicles. So in her current role, she's leading Exosomes team to harness the power of extracellular vesicles as a new class of therapeutic modalities. Andreia is a bioengineer whose research career has been dedicated to the study of extracellular vesicles and extracellular non-coding RNAs in the context of tissue repair or regeneration. So currently, she is a postdoc fellow at AstraZeneca, exploring EVs for improved delivery of complex new drug modalities. So today, their lecture is going to be about single-vesicle, single-molecule methodologies for EV analysis, application to quality control of EVs from the drug discovery to the manufacturing stage. So before we start, I'm very curious about Olga and Andreia, and I'd like to ask Olga to introduce herself a little bit more about her background, and then after that, Andreia, can you tell us a little bit what you've done prior to this and your current role? What is the Evotec and so on? Thanks, Olga.
Thank you, Carolina, for a nice introduction. Thank you for the opportunity to discuss our manuscript, our scientific work today with you guys and introduce our latest finding. And I think it's very important also to talk about quality control of extracellular vesicles. My background, my academic background, and honestly say it does not actually differ much between academia and industry. We are doing science, and actually the science is everywhere as a science. And I started as a scientist in Germany. So my scientific journey was started in 2006 in German Cancer Research Center. And I was working in the group of Professor Peter Kramer on the modification of TNF family member receptor, CD95, so death receptor, and glycosylation of this receptor, and basically role in the signaling of this post-translational modification, and also modification of the killing capacity. Then after this, I joined University Hospital Cologne, the group of Professor Elke Pogesch von Strandmann, and I was working on exosomes and activation of natural killer cell response. And I also was working on different modification of those exosomes, how one can manipulate immune response. And during this time, I realized, so basically it was a time when exosomes came really to the to the developmental stage as a therapeutics already. Like it was this time when first publication from Jan Latval group and Matthew Wood came in the field, and we realized basically that we can use those guys as a therapeutics. And then the next step was basically moving to industry and develop the exosomes further, and there we were building the exosome platform together with the exosome team, which is actually presented here today in Nick and our colleagues from AstraZeneca. And it was actually work of multidisciplinary team, very, very great teamwork. So we, like in a couple of years, we developed a lot of I would say scientific impactful findings. And basically my work was mainly focused on RNA loading of the exosomes using gene modification, and also using post-isolation loading. And basically, Andreia, I think it's afterwards I moved to Evotec, and where I continue my scientific journey with exosomes, and we are developing exosome platform from basically very beginning, from discovery stage, where we actually look for non-modified exosomes and also for engineered exosomes. We are working also on the upscaling process. We have bioreactor team integrated into Exosome project. We are working also with all platforms which are existing at Evotec. We are using proteomic and transcriptomic platform, and of course we have GMP facility with us. But the very first step which we actually started with was the development of the proper quality control platform at for Exosomes. So this is main summary. Thank you. Sorry,
thank you so much, Olga.
Worried. Yeah.
No, it's a congratulations for the move from academic to industry. I think that's quite exciting. And Andrea, can you just give a brief introduction on yourself?
Yeah, absolutely. Welcome, everybody, and thank you for joining the session today. And thank you, Carolina, as well, for hosting us today, presenting the work that we have developed in the last few years at AstraZeneca. So from my side, I guess both me and the extracellular vesicle field kind of start growing more at the same time. I started my PhD in 2012 at I3S INEV in Portugal, under the supervision of Dr. Susana Santos, and we were studying the role of extracellular vesicles in the crosstalk between mesenchymal stem cells and immune cells in the context of bone repair and regeneration. So it was a lot about the natural functions of EVs per se. And during this period, I was able to join as well Dr. George Scalin's group at MD Anderson Cancer Center in Houston, Texas, to work a little bit around extracellular RNAs, either inside vesicles or secreted but outside vesicles as well. And this was a very cool experience as well for me. I finished the PhD, and in 2019, this was my very first contact with a work environment different from an academic one. And in 2019, I then joined AstraZeneca Nick Stecker team. I had the great pleasure of joining Olga and Elisa on the team they started building up together with Nick. And since then, I have been working mostly on engineering of extracellular vesicles, so looking a little bit from a different angle for our little buddies. And I've been mainly focused on RNA loading into the vesicles.
Okay, well, we're looking forward for your talk. Then this all the exciting thing. Really want to know. Now, before we start, I just quickly want to thank Horiba Scientific who's sponsoring this session.
Okay, let's start the discussion. And today, thanks a lot once again for the nice introduction, Carolina, and the opportunity to introduce ourselves. And basically, we are going to talk about our latest manuscript, but also we are going to talk about from in the context that how you can apply the technologies on each and every stage of the pharmaceutical development of EVs, extracellular vesicles. And basically, today we are going to. I cannot move to the next slide. Andrea, could you pass, please?
Yeah.
Okay, so basically, before we start the talk, as Carolina mentioned, so it was basically, teamwork and great collaboration led by AstraZeneca Extracellular Vesicle team and all the co-authors from AstraZeneca. So it's a really interdisciplinary team and led by Nick and also by all the people who were driven this project, and in very close collaboration with Nano FCM and Nanoview. And basically, I'm right now an employee of Evotec, but the project was done during my time at AstraZeneca. Could you pass by to the next one? So basically, just to refresh your mind, all of you know what is the extracellular vesicles, and basically here we are going mainly to talk about the small fraction of extracellular vesicles, which people used to call exosomes, with a size from 100 to 140 nanometers. And well, we know that basically they were discovered in 80s, and just until 2007 2011 they were really forgotten. And then during this time, basically it was realized that these vesicles are important to cell communication, could be engineered for the targeting and also for the cargo loading. And so basically it was a starting point for the development of exosomes as a therapeutic modality, but very beginning. And right now, during this time, this is basically rise in field, and also there is a lot of clinical trials registered, so about 250, and 30 of them are actually focused on exosomes as a therapeutics, exosomes of different origins. So it's basically could be naive exosomes, which are derived from the cells and just be the copy of parental cells and contain the effector cargo which produced by the cells. But also exosomes could be modified genetically, so meaning that the parental cells could be genetically modified producer cells, and those exosomes could contain the cargo, therapeutic cargo could be also, the surface could be decorated for the targeting, for example, of the specific tissue, or could be targeted, decorated with, for example, some cytokines on the cell surface to make them more efficient for the therapeutic use. Also, exosomes could be loaded after purification, and this is what we called post-isolation purification differently, loaded with the small molecules, loaded with a nucleic acid, and for example, hybridosomes. Andreia, I see you are here. So hybridosomes is a great approach, basically, when you use lipid nanoparticles as a donor of nucleic acid cargo, and basically you can just fuse or incubate those two together. And of course, this is all existing, but very good question when it comes to clinical stage or preclinical stage. How we actually control all of this? Because we know that exosomes by their nature, they are very heterogeneous, and this is really crucial for GMP stage product to understand how we can characterize it in a best reproducible and robust way. And here, so there is just a couple of approaches which are mentioned, and this is just the standard biophysical characterization, like size, distribution, concentration of these exosomes, also composition using flow cytometry and standard approaches like Western blot, and of course the structure and morphology visualization using electron microscopy, but also what is really crucial, and it's really individual in each and every case, the activity of those vesicles. So, and right now, when we are coming to the basically clinical stage, and we know that exosomes at the moment, or extracellular vesicles, they have no real guidelines for the regulatory. So basically, if you pass for the next square, then we will see it's in the text. Yeah, basically the exosomes are categorized as a complex biologics, and basically there is a recommendations, but there is no guidelines for the proper like development, formulation, and application. So far, it's all under the development, as it's a very new field, and this is of course very challenging product for the drug development, as we mentioned, because they are heterogeneous. They are very the purity is varied due to the different purification approaches which are existing in the field, and there is no optimal approach at the moment. And there is a challenges with the scaling up and also purifying at the same time. And batch-to-batch variabilities, this is a problem which we are facing for each and every product we are developing. And of course we still not 100% defined with the proper formulation and preservation conditions, and also stability time of this product. And we know that also, as a complex biologics, the mode of action of those products could be defined by several components. So basically the characterization becomes a very challenging when it comes to the clinical stage. And the most important for us in the field right now is basically to define the proper quality control parameters actually to be applied for this product. So, and for now, right now, what is recommended, so basically quality controls procedures are adjusted for specific analytics of exosomes for like ATMP products mainly, and biologics and combination. And for example, also if it's a complex or if it's a biologic, or we are looking for some of the pathways, so basically it should be applied for individual case and for the indication you're working with. So could you pass to the next slide, please? Yes, thank you. So basically, we are coming closer right now to the discussion, and basically each and every pharmaceutical project goes to the developmental stages up to clinical development. And we start usually with the process, the product discovery, process development, and then of course clinical development. And during each and every stage, quality control plays a crucial role. It's less complex, less I would say less maybe less defined in the product discovery stage, but should become more and more defined to the next stages. And for example, when we think about naive or engineered vesicles, when we think about maybe mode of action, we of course show all of this during the product discovery. We show proof of principle for or mode of action for this modality, and this is done, of course, in the cell-based assay designed for this product. And of course, we think about the safety and efficacy, and of course, optimization of this product. But then we come to the next stage, to the process development, and here we are becoming more and more strict, and we are working, of course, on the optimization of the production and purification, which is really crucial. And by the end of this product product development stage, we basically come already with a protocol which could not be, or which should not be, changed in the clinical development stage. And this is very well defined process. And by the end of this process development stage, so basically we need to come with a process characterization and product characterization. And this is something which we are going then to use for the clinical development and real characterization already in phase 1, from phase 1 to phase 3 studies. So here we highlighted a couple of publications which came also from AstraZeneca side, and this is a work on which was done on optimization of the cargo loading and also on the process optimization. So basically on, for example, anion exchange chromatography application for the purification of extracellular vesicles. We also have done some work on the safety and toxicity of these products, of the naive extracellular vesicles. And so we are still thinking about the proper quality control of the product, and we are thinking how to develop these processes for the next steps. So Andreia, could you please pass by to the next slide? And here just an example what we are doing with our team, who is also present today. Thank you guys for joining. With extracellular vesicles prep and with the small extracellular vesicles. And as I mentioned, this is different from case to case. We start usually with a biophysical and morphological characterization, and this is usually concentration, size distribution, and also phenotype and EV marker presence. And this is our very, very basic, and this is as a must for each and every prep which we are using. And then depends on the next steps what we are going to do. So we call the first part as a standard QC, but the rest of the characterization it comes, and this basically depends on the purpose we are actually using these vesicles. Either it's optimization of purification or cargo analysis, which we would like to load, or we would like to define the mode of action, or this is something as a stability, or this is something as a GMP grade production. So in this case, of course, the characterization is complete, and safety and biodistribution and efficacy, those vesicles go usually through all the characterization steps. And as I mentioned, the dark blue is a very, very standard, and the white is a very specific. And in this case, so something which we apply depends on the purpose of our work. And usually for the EV composition analysis, what we usually use is standard approaches like Western blot, qPCR, but we also have unique transcriptomic and proteomic platform, which we are also applying on the regular basis when we are working on, for example, purification optimization, and when we are looking for the mode of action of our vesicles. But in this case, of course, you generate a very like a big data set, and you need to think about real fingerprint, which you use usually if you think about the purities, and you can definitely define the markers which you need to look for. But also you need to check what your formulation contains and how reproducible basically this production of extracellular vesicles and QC. And of course, the next step is the safety and toxicity and efficacy analysis of our vesicles, which we usually test first in the cell-based assays, and then we will go to in vivo model. And before we start any cell-based assays, of course, we do endotoxin analysis, and this is a must before you do any cell work. So basically, this is just an example, and now this is a time to start our discussion. And if we can just move to the first slide, and this is our publication, which was officially online in August this year. And yeah, this is about quantification of protein cargo loading into engineered extracellular vesicles. So we are talking about a special case, but all these technologies which we are going to discuss today, they could be applied to the different steps of extracellular vesicle characterization. And all the authors, you can see them on the right side. So basically, it's like in the order. So Andreia, Elisa, Anders, Shabi, Niki, Elisa, me, and Nick, and also Ben, Aidita, and George. So thanks everyone for the great and productive teamwork. So, to start with, yeah, basically the goal of our work, and of course of this manuscript, is to produce engineered extracellular vesicles with loaded with the therapeutic proteins on the surface of exosomes or loaded intraluminally, and to basically develop this to develop this modality. We were thinking about the selection of the EV sorting proteins, and then basically EV sorting proteins which could be fused to specific proteins of interest. And then, of course, the second very important part is to determine the efficiency of this engineering and cargo loading. And in this case, we were evaluating the different approaches. So we start with a very standard Western blot analysis, which everyone can do in the lab. And this is, of course, a bulk approach. And then we came to Nanoflow, cytometry, ExoView, and also single-molecule localization microscopy. And these three technologies, the last three technologies, they have single-vesicle resolution, but unfortunately not a single-molecule resolution. And depends on which stage of your product development you are. If it's just a scientific or discovery stage, so basically you probably don't care much about how many molecules you load per vesicle. But if you come further and further, you need more defined quantification of your product, and then definitely single molecule microscopy might be of a very good help in this case. So could you move to the next slide, please? And yeah, basically this is a workflow of the paper. And as I mentioned, we started with a selection of the EV sorting proteins. So the proteins which are highly enriched in exosomes, we started to check first in the public available databases like ExoView, ExoCarta, and Vesiclepedia. And of course, we checked how many publications were associated with these proteins. And basically we looked for CD47, syntenine, AppMap, tetraspanin 14, tetraspanin CD63, and also modified tetraspanin CD63 and CD1 with a transmembrane domain of the PDGFR beta. So, and in this case, we were thinking about also the size of those proteins. So they should be not more than 50 kilodalton to basically facilitate the engineering and also expression of these proteins when they are fused to the target, so to the target protein. In this case, we were using GFP, but also GFP with a MIG-tag, but also it could be different protein of interest. And then we basically also checked these proteins which were used as a EV certain proteins. They are not only small, but also non-toxic and non-immunogenic and not interfering with the EV production in the cells. So, and what we have done afterwards, we clone those constructs with as it's drawn here. So we designed C or N terminal fusions of these proteins. We transfected transiently XP293 cells, collected the conditioned media, and performed standard differential ultracentrifugation, basically to purify the small extracellular vesicles, which we call exosomes. And then all the vesicles went through the very standard quality control procedure, and in our case, we defined it for ourselves. We would like to see the proper shape and morphology of those vesicles. So we perform electron microscopy analysis, we perform nanoparticle tracking analysis to define concentration of those vesicles and also size distribution, and we checked for the exosome markers using Western blot analysis. But this was just a starting point, and then basically we checked, of course, for, as it was a GFP fusions of these proteins, we checked first for the GFP expression and enrichment in extracellular vesicles and cell lysate. And we tested, of course, you can read the publication, we tested several antibodies. Here is just a representative Western blot. And what we found out here, so basically that all our fusions were expressed in the cells in different level and also were enriched and found in exosomal fraction. So we then also find, you can see visually that there is like 3 main proteins like T-span 14, CD63, and also CD81 fusion to PDGFR. They were highly enriched, and this was confirmed also for using the semi-quantitative densitometry analysis. And basically what we found that, okay, there is these 3 proteins which were highly enriched, other proteins were enriched less. And for example, CD47, syntenine and UpMap, they were enriched and less great. But what was interesting that, for example, UpMap was enriched highly in the cell lysate, but completely found at completely less extent in extracellular vesicle fraction. Of course, this is a nice visual data, but we cannot really talk about real quantification of this enrichment and also distribution of this GFP fusion within the extracellular vesicle, so single vesicle fraction. So basically, then we start with a more deeper characterization. Andreia, all yours.
Yeah. Exactly. So at this point, we were not only interested in understanding which were the EV sorting proteins that allowed the loading of our cargo of interest in the vesicles, but how efficient those proteins were in doing so. So we started our analysis using a single vesicle analysis technique called nanoflow cytometry, which work principle is very similar to conventional flow cytometry, but it takes benefit of low-speed fluidics and site scattering and fluorescence-triggered event detection for nanoparticle identification down to a size of 40 nanometers, which means that indeed we are in the space of single vesicle resolution when analyzing our samples. In this work in particularly, we look not only to the GFP cargo, fluorescent cargo of our vesicles, but we also labeled our total EV population with the interluminal cell trace far red dye. And according to our results, we could find that more than 85% of our detected particles were cell trace far red positive, meaning that we actually have highly pure EV samples that we were working with for the different EV sorting proteins tested in our work. Of course, then we looked for the percentage of positive extracellular vesicles carrying the GFP cargo, and then again we confirmed the EV sorting, this set of EV sorting proteins that we had seen by Western blotting that led to the highest enrichment of GFP in the vesicles. However, very interestingly, at this step, we found that actually only less than 50% of our vesicles were successfully engineered with the GFP cargo, which was a feature that was completely missed by our previous Western blot analysis. We further explored this specificity of nanoflow cytometer for this kind of analysis, and for that, we actually compared the levels of GFP when it's overexpressed in cells in its free soluble form, meaning that it's just passively loaded on the vesicles, relative to our worst and best performing EV sorting proteins tested at the moment. And when you look at our Western blotting results, we see that actually the GFP levels found for overexpression of free GFP protein are actually quite similar to the GFP levels when it is fused to the tetraspanin 14 EV sorting protein. However, when we do single vesicle analysis by nanoflow cytometry, the scenario changes completely. And now we see that the free GFP, we see that a much lower percentage of vesicles actually carry the free GFP when we compare it with vesicles successfully engineered with the tetraspanin 14 fused to the GFP cargo. So we basically believe that our Western blotting is capturing probably secreted free GFP that is contaminating our EV samples, but nanoflow cytometry was definitely capable of dissecting this little detail that we had missed completely just by a Western blotting analysis. Next, we were also interested in understanding what was the distribution of our GFP cargo in the different tetraspanin positive EV subpopulations. And for that, we used Exosomes assay. And in this case, we used sheeps functionalized with anti-CD63, anti-CD81, and anti-CD9 antibodies printed in specific spots on this sheep. The sheep was incubated with our EV samples, and then we immunostained them with a cocktail of anti-CD63, anti-CD81, and anti-CD9 antibodies. And the purpose of this was to capture the whole population of EVs that was specifically captured on the sheeps that we use, and that's what you see in the red dots here in this figure. And in this technique, we are also working at the single vesicle resolution, and then we went to look for the percentage of positive GFP positive vesicles in each one of the different capture antibodies. And what we see is that there's a clear heterogenic distribution of GFP cargo across the different tetraspanin spots. And we see, for instance, that CD9 positive vesicles were the ones carrying showing the lowest percentage of GFP positive EVs. But on the other hand, on the CD63 spot, the vesicles that were previously engineered with CD63, almost everything that was captured on the spot was GFP positive, so carrying the cargo of interest. And the same for the CD81 spot, where we see that upon overexpression of CD81 transmembrane domain with PDGFR, we actually see that 80% of these vesicles were carrying GFP, which is kind of expected, right? Because we are overexpressing the proteins that are being detected on the sheep. So definitely we take advantage of the antibody epitope affinity here. On the other hand, when you look to the other best EV sorting tested proteins, we see that we have around 40% of GFP positive vesicles, either CD63 positive, either CD81 positive. Of note, very interestingly, we did the exactly same experiment using condition media depleted of larger vesicles, but without further concentration of the small extracellular vesicles, and we got a very similar trend in our results. So it's a very interesting feature for less complex analysis workflows. At this point, of course, we were interested as well in understanding not only how many vesicles were carrying GFP cargo, but also what's the level of cargo that each vesicle is carrying. And for that, we went to look for the mean fluorescence intensity of GFP, either by nanoflow cytometry and also by the Exosomes platform. And in both platforms, we could see that tetraspanin 14 and CD63 EV sorting proteins were the ones that led to the highest enrichment of GFP cargo in our vesicles, and particularly for the CD63 positive vesicles, but this was also seen for CD81 positive vesicles and CD9 positive vesicles in our samples. But at this point, we had already an idea, okay, how many vesicles are successfully engineered? We have an idea of the relative enrichment of GFP in these vesicles, but we still didn't have an actual number. How many molecules of GFP do we have in our vesicles? And in order to know that, we implemented a single molecule localization microscopy workflow, which is a simplified TIRF workflow, where basically basically, we image our vesicles taking advantage of high numerical aperture objectives, and we get images something like this on the left. And basically, what he did in order to determine the amount of GFP molecules that our vesicles carried was to do imaging processing of the images that were acquired, so that we knew exactly the location of where each spot, GFP spot, was localized in our images. And we also compared the photo-bleaching traces of these GFP spots with the photo-bleaching traces of recombinant GFP molecules that we were using as a reference in this work. So by doing this comparison, we were able to detect which spots, GFP spots that we see on the image actually correspond to single molecules, and if they belong to the same vesicle or not. So by this technique, we are operating with a resolution level at single vesicle, but also a single molecule. And very interestingly, when we did this approach, we actually showed or have seen for our best performing EV sorting proteins that tetraspanin 14 leads to the biogenesis of vesicles carrying actually very high copy number of GFP molecules, more than 100 molecules per vesicle. On the other hand, CD63, yes, it's a very good EV sorting protein to have high percentage of positive vesicles, but you see that the majority of vesicles actually carry less than 100 molecules per nanoparticle. So by doing a quantitative analysis of our different EV sorting proteins, we actually verify that tetraspanin 14 can lead to an enrichment of about 150 molecules per vesicle on average, right followed by CD81 fused to the PDGFR transmembrane domain with around 100 molecules per vesicle on average. So summing up about what we talked to you today, it is important to understand that it is extremely important that we have adequate and complementary techniques to characterize our EV samples, our EV preparations, because many of the techniques that we currently use alone, they can have a lot of drawbacks and disadvantages that might lead you to miss some features of your samples that you can definitely capture when you use complementary techniques. And definitely we have shown that bulk techniques that basic techniques that analyze EVs in bulk, like Western blotting, are very good in probably early stages of EV drug development, where you are interested in doing a very fast screen of the product that you are developing. But this technique completely lacks resolution and sensitivity to actually capture the heterogeneity that we see on our vesicle preparations. And in this scenario, Nanoflow cytometry and Exosomes assay that we use in this work actually have much higher sensitivity and resolution for this kind of analysis. And this is probably the stage where you want to be when you start developing your product further, because it will allow you, for instance, to design better dosing schemes for your in vitro and in vivo studies, because you know exactly what your vesicles carry. And this is the same features that we see for single molecule microscopy, with the advantage that single molecule microscopy was actually the only technique that we tested in this work that gave us the actual cargo copy number that we carried on our vesicles. So very high performance of this technique to achieve that feature. And that is probably the stage that you want to be, where you are producing your vesicles at GMP conditions to go into clinical trials, where you actually need to have a very good definition of all the cargo or of your cargo of interest on your vesicles. So it is important to understand that at the moment, we don't actually have compulsory or mandatory guidelines that dictate what is the QC that we should do for our extracellular vesicle preparations at different steps of drug development. But definitely, we believe that the discussion in the field and awareness to the importance of doing very thorough characterization and quality control of the vesicles is extremely important. And I have focus working groups have definitely contributed to raise the awareness for this important question. And in the meantime, while we don't have or we don't reach more consensual guidelines to achieve quality control of EVs up to the clinical application, it is up to each one of us to try to do the most comprehensive characterization of the vesicle preparations that we can within reasonable limits, of course. Finally, before we finish, we just would like to thank everybody in the team at AstraZeneca contributing to the development of the EV work, in particularly to Nick Decker that leads the team, and also to all the authors of the main work that we presented here today. So Anders, Xavier, Nikki, Elisa, Christina, Ben, George, and Aditya. We would like to acknowledge as well the teams of cell therapy assay development and chemical biology and proteomics at Evotec for the useful insights on the development of quality control pipelines for extracellular vesicles. And thank you all for joining us on this discussion today.
Thank you so much, Andrea and Olga. Wonderful presentations, and that's really quite interesting data that you have there. I wonder if everyone wants to have a one-to-one question. Please go ahead, raise your hands if you can, or you can also write it down on the chat box. So I think I'll start with my questions then. So that's quite interesting that you only have like 50% of the vesicles that has the GFP. So would that you think correlate with which these vesicles coming from, whether they are different type of vesicles, or do you have some sort of like additional insight on that?
You can start if you want.
Yeah. So basically, I think we both can come this. Of course, I mean there is couple of proteins which are published to almost be enriched by 100%, and some of them like developed by Kodiak. But so in our hands, with our experience, due to the nature of extracellular vesicles and so basically the pathways which we are actually produced, you cannot reach, of course, 100% of the vesicles to be positive. But so there is many pathways involved. Of the best, very well characterized SCAT pathway, but also there is other pathways. And in principle, I think it's a very good, it's a very good enrichment, 50% of the positive vesicles, and especially if you have 100 copy per vesicle. So basically, what is right now missing in the field and what is highly needs to be developed is basically the technology where you can capture your vesicles and then release them and have very well defined population of the vesicles, and this would be the best case. However, the standard techniques which are existing right now, so if you use immunopurification approaches, so the capture the capturing capacity is about like at the end 1 to 2%. So you cannot reach 100% of your population being positive for even if you do purification after the loading. So basically, this is something which is missing right now in the field: the proper purification when you can enrich your subpopulations. But also 50%, I think it's reflecting the natural situation, and this is what is existing.
Yeah, exactly.
We have to deal with it, right?
Yeah. Other groups have reported similar levels of successful engineering of the vesicles for proteins similar to the ones that we used. And just going back to one of the comments that Olga said, so ideally we would want to capture these vesicles from our preparations to have them, let's say, if we want to do our in vitro or in vivo or even clinical trials, right? And this might be easier achieved for the vesicles where we actually have the cargo on the surface. But if you think about cargo that you want to load intraluminal, then you have an extra layer of difficulty in actually being able to capture the ones that carry your intraluminal cargo of interest. And this is an extra layer of difficulty as well if you go for RNA, where you probably don't have so many tools for capturing those vesicles as you have in the protein space.
I see there's a couple of questions coming up. Joel, can you turn on your video and ask your questions, please?
I'm afraid my video doesn't work, but thanks a lot, both of you, for an excellent talk. We do have a question around the efficiency of loading, since some of the proteins that you use have an N-terminal or a C-terminal on opposite sides, whether you've ever looked at differences, whether you're displaying something for the surface or for luminal cargo.
Very good question.
Yeah, I'll let you go on this one, Olga.
I mean, of course, it's not the all proteins which we tested, right? So we screened quite some. Yes, we definitely tested different fusions, different modifications. And for example, as you can see in the very first slide, we were using CD63 construct where we were introducing the cargo into the tetra loop of CD63, but we also were playing around with this, and we were doing, of course, optimization, and we were adding also some fusions into C-terminus and also in N-terminus. And for example, it's known also from the literature, not only from our experience, but we repeated this basically that, yeah, you can introduce these modifications, but not all of them are tolerable. And for example, N-terminal fusion to tetrasponins is usually not very successful in enrichment and also in expression. But for the other proteins, you usually test different modification before you start, of course, with the next steps. So, did I answer your question?
You did. We just have to test it. But thanks a lot. It makes sense.
Right, Leonie, please go ahead.
Yeah, hi. Thanks for the great talk. I was just wondering why you not looked at the CD9 for the GFP engineered EVs that you were looking at, because that the recent work also shows that especially the small vesicles are more likely to carry CD9. And you were looking at the exosomes, so I was wondering why that was not in your figure there.
As Olga just explained, so this all has to do with so we started by screening a lot more proteins than what we actually ended up wrapping up for the final publication, right? And this has a lot to do with Olga just explained. So we did had CD9 in our initial test, but it didn't perform so good as the other proteins that we included in the panel that we presented today. And of course, this has a lot to do where you do the actual fusion protein with the tetraspanin, with how stable that fusion is, how well it is expressed in your cells. But in our hands, it was definitely not one of the best performers, and that's why we left it out.
Yeah, thanks. But that's also interesting, right? To know that it was not good, not well performing. So that's also good to know. It's a good answer for me as well. So thanks.
Okay, someone else. So I have actually another question. So when you look with the microscopy, and there is also a difference of the molecules number by microscopy assay in comparison to the copy number. So I just want to know, like, what is the technique that you use to look into the copy number of the GFP in the vesicles? Because there's like you understand my question, sorry. There's like 100 molecule of GFP was shown by the microscopy, but then there's a graph that you're saying is a copy number that is 150. So can you give a little bit more detail on the experiments you've done for that?
Yeah, so actually on the single molecule microscopy approach, you get the data that you actually get is a distribution of the different vesicles that you have on your image and how many molecules each vesicle carry. And if you remember the plots that I showed, you basically have a distribution with vesicles carrying actually a quite low number of molecules, with vesicles carrying a much higher number that can reach up to 150 molecules per vesicle, right? So, or even more. So then when you average that for the whole population, you get to the 150 GFP molecules per vesicle, but basically you get the whole distribution. a cargo on your vesicles. Whether this is related with biogenesis pathways, for instance, if it is different subpopulations of vesicles for markers that we didn't test, or even for CD63, CD81, and CD9 that we didn't test at single molecule level. These could be very good questions and could give us very good insights about the biogenesis of these vesicles once you actually manipulate the system doing overexpression of this kind of proteins, right? We didn't do this analysis, but that would definitely be very interesting to further look into.
Yeah, but in that vesicles, it's not only just GFP anyways, right? So there's actually like many more molecules in that.
Yeah, exactly. Yeah, and at this point, then again, we don't know when we do the overexpression of these proteins, we don't know how much we influence the biogenesis pathways of the vesicles that we isolate, right? So it's definitely possible that when we do an overexpression of a certain protein, we end up enriching our preps with a slightly different subpopulation that happens to carry as well other types or other amounts of other proteins, and that definitely needs to influence how much you can pack, because the vesicles have limited size, right?
Yeah. That actually, in terms of biological point of view, I think that's very interesting. How many proteins can you pack inside that small vesicles?
Yeah, exactly.
Another question from Christian. Can you turn on your video, Christian?
Yes, can you see me? I hope this works now.
Go ahead, please.
So very nice work. Thank you for this clear presentation. And as maybe a naive question. So if you perform this overexpression of these engineered proteins, and you showed nicely that, for instance, tetraspanin 14 can be detected with up to 100 molecules per single vesicles. So do you have any or did you do any observation that also the vesicle sizes then increased by simply calculating tetraspanin, or this tetraspanin 14, I would assume account for 2 nanometers in diameter approximately. And if you have up to 100, these are then 200 nanometers. This is already the size of AV. So did you observe something like that?
Olga, would you like to answer?
Yeah, we can do it together.
Yeah, so according to the analysis that we did by nanoparticle tracking analysis and with a nanosite instrument and by nanoflow cytometry, we didn't find a significant difference neither on the mean, neither on the mode of the vesicle populations that we were able to detect by these two techniques. We also didn't see that on the ExoView assay. So if that happens, we were not able to capture that in our assays, in the techniques that we tested.
But it's a very good question, and especially that it's C-fusion and we got the cargo inside and it was GFP, but mainly tetraspanin is located on the surface, right? So maybe this is not that crucial if you have like a luminal loading of the bigger proteins in this case. But also like particle number, we observed that the particle number changed, not dramatically, but yeah, we increased the particle number upon overexpression of some of them of tetraspanin and CD63, for example, but not changes in the size we observed, but basically maybe we were not able to detect them at this point.
Okay, thank you.
All right. Anybody wants to ask the last questions? We are approaching the end of our session. So if not, and then I'd like to close this meeting. And thanks so much, Olga and Andreia. Great presentation, and also giving an insight on what we need to do in terms of QC, especially if we want to do for therapeutic application.